Um, my personal thank you to all of you that are coming. This is the sixth such occasion. I think it is the second or third linking in with metallomics. At the end of this morning's session, Matt, our senior postdoc, will be talking to you about applying physiological oxygen using Baker-Ruskin, BMG lab tech, and what we do with metallomics. Yeah? That chap over there, who's bald like me, is very nice. I'm just picking on him because that's Jörn Steiner, a former PhD, and he said something really nice to me. He said, Giovanni, even though you look a little bit, he said, you still have the same enthusiasm that you had 15 years ago. So that made me feel good. Anyway, today, what I'd like to do is to say thank you, obviously, to the British Heart Foundation, Heart Research UK, definitely Baker-Ruskin and Andrew Skin, long lifetime colleague and friend. Agilent, who are not with us, they used to be Luxol, but they took over Luxol. I'll share some of that information with you. Clearly, BMG LabTech, Robert Mann, our friend, Catherine Moore, and Oliver, who's going to talk to us. And finally, I'd like to thank LabTech for helping us so kindly today and setting this all up. So in my first slide, what I'd like to share with you may have seen this in the literature. What we have here, let's start on the right-hand side. We have the air we breathe at around 21 kilopascals. For today's talk at sea level thereabouts, this is 21% oxygen. As it passes into the lungs, it's around 20 kilopascals. In the alveoli of the lungs, it's around 14 or 13. Depending on the organ, be it the heart, the liver, the kidney, you could have anywhere between 5 in the coronary circulation to 30% in the lung. And in tissues in the microvasculature, it can be as low as 1 to 5. And within mitochondria, about 0.5 to 1.3. So the take-home message, if you get nothing out of this slide, is this. If we do experiments in air in a standard cell culture incubator, which we have upstairs, I will show you data that the intracellular oxygen level measured by us using at that time MitoExpress intra from Luxol, now under Agilent, is somewhere around 5% if we're under physiological oxygen, and we can set that in the Baker-Ruskin workstations, and I'll show you. But if we do it in air, we're sitting up here. But when we look at the organs we're interested in, the heart, the kidney, that oxygen level on this axis here in kilopascals per percent oxygen from 0 to 25 is significantly lower. When people study hypoxia, and this Thursday will be a hypoxia session that Krista can mention, put on by Baker-Ruskin, they take a cell from air, room air that we're breathing, and they move it down to hypoxia less than 1% or 1%. That's an acute reduction in oxygen. I'll show you data why when we look at physiological normoxia, we have to adapt cells for a longer period of time. So that's sort of the background intro slide I wanted to share with you. I'm only going to show you one picture from Tom Keeley's in my review in Physiological Reviews published in 2019. The title of that was Defining Physiological Normoxia for Improved Translation of Cell Physiology in vitro work to animal models and humans. Look just here, we pop here, this is the coronary artery in the heart, the human heart, and you will see that in the coronary artery, it's around 5 to 7 kilopascals. And Tom, what he did, he went through all the literature related to different regions of the myocardium to find the actual published measured values. And these are all for the kidney, for the lung, for the heart, for bone, for cartilage, for stem cells, sitting in this physiological review if you'd like to study it. This is important for anyone that works in cell culture. When we have the cells in a microtiter plate and we put them in an incubator, we can measure the pericellular oxygen level here. And if we were to set this pericellular oxygen level, for sake of argument, at 5 kilopascals oxygen using a Baker-Ruskin workstation, as I'll show you, then a HeLa cell is shown here would roughly have the same oxygen as ambient. The umbilical vein endothelial cell that you see with the arrow here is around 3.7 kilopascals. But if we take a contracting myocyte, note that if the ambient oxygen or pericellular level is 5, that cell will be hypoxic. Why? Because there's no blood flow. These are static monolayers. So it's critical that you understand the metabolism of your cell. And it depends on the cell that you're studying that will determine the absolute intracellular oxygen level. And I should have said the x-axis is showing attomoles per cell per second of oxygen consumption in that cell type. And the review gives you more information. Okay, this is what you're going to see this afternoon. These are two colleagues. This is Salil, this is Tom Sheely, who's now working with Peter Ratcliffe in Oxford. And this is one half of a scientific workstation. They have these sleeves, you can put your arms in. The important thing to show you is that there's a little microscope provided by Etaluma, a screen, and here a little rack where all the cells are cultured. These cells are adapted within this workstation throughout their life in an experiment. They never exit the workstation into a laminar flow hood where you're going to trypsinize, feed the cells. All procedures are done within a defined oxygen. And on the lower panel, we show you two sides of such a scientific workstation. This side is gas at 5% oxygen or 5 kilopascals, this at 1%, and we can have cells from the same culture, the same blood vessel, at 1% or 5, and then study. Here I just show you a tri-gas incubator where hopefully you can see later the use of condensate units where you can have cells that have been treated in here, removed from here, left at the right oxygen level, and then put into a tri-gas incubator. Then when you want to study them again, you can move this back in through the central port, back to the oxygen level that you were interested in. So what we're trying to do is set the oxygen and the redox phenotype of these cells long term. Now hopefully that'll become clear more so in the next slide or two. Okay, that all up. This is when we worked with Concari and at that time Luxcel. What we have here is the MitoExpress intraprobe signal, for sake of argument, and on the x-axis the percent oxygen or kilopascal. As you lower the oxygen from air in this BMG plate reader that you'll hear more from Oliver, and you have cells loaded with MitoExpress intra, you can see that the signal increases as the oxygen is lowered. So we're able, in living cells that have been adapted to a fixed oxygen level in the workstation, to move them rapidly, 96-12 plate, into the plate reader, which has an oxygen control unit. And we can then determine the intracellular oxygen level in living cells in the presence or absence of a stimulus. And this, as you'll hear later from Oliver, has an injector, so you can inject drugs or inhibitors, etc. Now, let me just take you, I'm going to take you through all of this, but I'll walk you through the slide. If we have endothelial cells in room air adapted, and we move them into the plate reader, which is at room air, you'll see that the black is the cytosol, the squares are the medium, and we can measure the oxygen content based on the MitoExpress intraprobe in room air. We can lower the oxygen, and you see it rapidly over 2 hours, drops to around 4 or 5%. This is not sufficient to get a redox phenotype of physiological mammography. In vivo, with a blood vessel, the blood has 3.7% oxygen with blood flow. Note that in a monolayer that's static, there's a gradient, and hence at the cell monolayer surface, the oxygen level could actually be approaching hypoxia. And when we looked at intracellular oxygen in air, we saw that the intracellular oxygen concentration was around 16%. If we put cells at 3.7%, which was measured in vivo, we underestimate the actual reported value. But if serendipitously we use 5% oxygen, we achieve an oxygen level in the cell that represents the level in vivo. Really, really important. Remember what I showed you in the slide before. When you take cells, like here, and you put them to 5%, or if this were hypoxia, what's the issue? And that, I hope, comes up in the next slide. What's shown here on the y-axis is protein or RNA expression. This is HIF1 alpha RNA, and these are the downstream targets of HIF1 alpha protein. This is intracellular oxygen. If we lower oxygen from air to around 5%, we get an immediate increase in messenger RNA for HIF1 alpha, hypoxia-inducible factor. And that stays elevated for But note that the downstream targets of HIF, if target proteins are upregulated for days. So when you do an acute lowering of oxygen, yes, that's hypoxia, but that's not the redox phenotype that we wish to study when we're looking at redox biology. So bear that in mind, and I'll show you actual data in subsequent slides. Heavy slide. We're interested in redox signaling with Richard, and we're interested in this pathway, KEAP1 NRF2. This is a redox-sensitive transcription factor bound to KEAP1 in the cytosol, and under normal conditions, ubiquitinated and broken down by the 26S proteasome. So it's continuously being broken down. When we have oxidative electrophilic nitrosative stress in the case of Johann, this transcription factor and its association with KEAP1 changes its conformation. It now stabilizes NRF2. It can no longer be ubiquitinated. An endogenous accumulation of endogenously synthesized NRF2 binds to an antioxidant response element and turns on antioxidant defense genes. I won't go into the detail of that. So if we want to study this pathway under physiological normoxia versus air, we need to have our cells adapted for at least 5 days to a given oxygen level in the workstation. Whoops, sorry. What you see here, and it's not important that you know everything, this is a target of NRF2, a protein called hemoxygenase, plotted as relative to alpha-tubulin, a lobulin control in this immunoblock. All I'm trying to get across is one message. In passage 1, 2, and 3 of these cells, where a passage is 3 to 5 days, if they're left in air and you look basally, very little happens. But if you stimulate with an inducer of NRF2, you get a robust increase in hemoxygenase expression. If you have them for 2 weeks in air and then drop them to 5, you markedly attenuate that induction. If you go air, air, 5, and back to air, you regain it, and so on. If you go 5, 5, air, and back to 5, you lose it. That is telling you that the cell under a redox phenotype of 5% oxygen is not showing the same sensitivity to this NRF2 inducer, or for that matter, to an NO donor or other agents. And we have published this in this paper with Sarah Chappell as the primary author, and Richard in 2016, that we know that this is due to oxygen affecting the expression of a negative regulator of this enzyme. So if we take cells at 5% oxygen for 5 days, we transfect them with scrambled RNA, and we look at this negative regulator in the absence or presence of an NRF2 inducer. You can see that the protein level is here, but we cannot induce hemoxygenase. If, however, we transfect with siRNA, we silence the negative regulator, then you can see that even at 5%, we can rescue the expression of HO1. And that's telling you that in air, the negative regulator is upregulated, and in physiological normoxia, that regulator is not overexpressed. And I think, heavy slide, hyperoxia, artifact, this transcription factor can go to the nucleus, turn on most genes. But when we come to normoxia, this transcription factor can still translocate to the nucleus, but it now does not turn on these classical NRF2 genes because of the inhibition by the negative regulator BAH1. But genes involved in glutathione biosynthesis seem to be independent of changes in oxygen. And we proposed in this paper that it is these particular antioxidant genes, the glutathione-related genes, that are regulating redox defenses in vivo against oxidative stress. This is recent work from Gabby, Matt, Richard, myself, and Tom. These are now in brain microvascular endothelial cells. And what you can again see here very quickly is that if the cells are in air, very little HIF, 3 cultures, but at 1% there is HIF stabilization. Here's the data to illustrate that. More importantly, an antioxidant enzyme catalase, which scavenges hydrogen peroxide, is lower expression at 5%, as is glutathione, again telling you that the cell under 5% oxygen is not under hyperoxic stress. For those that are interested in function, we look at cell count. The cells actually proliferate more slowly in 5% than 18. So there are all kinds of phenotypic changes in adapted cells. I'm going to skip that. This is for Jörg Steiner. He's interested in nitric oxide, we're interested in nitric oxide, and there's a thing called proximity ligation assay, where you can have the close association of two proteins. And what we have here is cells in vehicle under 80% or stimulated with histamine. I can barely see it. There are more red dots here, okay? If you don't believe it, they're here. Right. And what that is, that the association of this phosphatase with enos is markedly enhanced in these cells. And if you read this paper, you can see that you can modulate NO bioavailability and also the dephosphorylation once it's activated. But the simplest message for scientists, and I often when I teach my students say, taxi drivers, bus drivers, or your mother or father, is this. We've got endothelial cells, this is work from Tom. We're looking at the cell viability, how do they live? And we're challenging with a compound, ionomycin. It punches effectively holes in the cell membrane and allows calcium influx. So it's a calcium ionophore. What you see here, if cells are in air, they die beautifully. But if we adapt them to 5% oxygen, they're protected at a given dose of ionomycin. So this is, in real physiology or clinic, this is calcium overload in the heart or in the vasculature. This here, we're staining for if a cell gets damaged, phosphatidylserine groups can extravasate, and they can be tagged with an Nexen. You can see here Nexen patch staining, very prominent, markedly protected if the cells are at 5%. So the cells are protected, the cells have increased NO bioavailability, the cells show certain proteins involved in getting calcium out of the cell altered. And if you read this paper, this pump, the circle pump activity is actually inhibited in cells cultured in air compared to physiological normoxia. I always show this slide because, again, it's a bit of work that we did with Ben Forbes in Mudway, Richard and I. And what it shows here is epithelial cells taken from the lung, and we're measuring, so boringly, glutathione, the key intracellular antioxidant in nanomoles per milligram protein. And this is in air standard incubator. And then we're challenging those cells with nanoparticles to see, do the nanoparticles kill the cell? And if you look at this, these cells are highly resistant until you reach a very high dose of nanoparticles where you now damage the cell and lower glutathione. Take the same cell and put it in 13% oxygen, which I said at the beginning of my talk is what's measured in vivo. And what do you see immediately? That under basal conditions, glutathione drops from about 60 to 30. Why? Because the redox stress is significantly less in a cell under the appropriate physiological normoxia. I always make the joke that this paper could have gone to Science, go forth, go into the clinic, nanoparticles, harmless. This one we sent to the New England Journal of Medicine. Be careful about nanoparticles. They're killing your cells if you have the right oxygen phenotype. For those that found the lecture too fast, too diffuse, please do take the time to look at the Physiological Review in 2019. There's everything you want to know about oxygen bioavailability, oxygen metabolism, oxygen level in different organs, and clear guidelines how to culture your cells under the appropriate oxygen levels. Life would never happen without the team. Here's Tom Keeley, sitting here, Richard, I think there's Giovanni, there's Sarah, this is Emily, and here we've got Emily and Matt actually in the chamber. This is Gabby who did the brain work, and there's Tom and Sonia, and sometimes we have a good time. So with that, I'll stop. Thank you very much.